Good afternoon. The topic for today's PG activity is tissue culture. Before going to the topic of discussion, let us brush up a little bit upon the history. Ross Harrison in 1907 was the first one to experiment in tissue culture, and he used the hanging drop method to culture a frog neuron. Although his efforts were great, the experiment was not very successful. In 1912, Alexis Carrel built upon his work and successfully cultured small sample of 18-day-old chick embryo, and he became the first scientist to culture mammalian cells in vitro. And coming to famous HeLa culture, which was the cervical cancer cells from Henrietta Lacks, which were further used for the creation of the cervical cancer vaccine, which we know presently, and it was done in 1951. Today, tissue culture is used to investigate diseases and study the pathological cascades. This was the timeline of the development of tissue culture, and it was developed through various experiments. So what is tissue culture? It is a method of biological research in which fragments of tissue from an animal or a plant are transferred to an artificial environment in which they can survive and function. So it is basically a blanket term or a generic term which is used for in vitro cultivation of cells, generally consisting of heterogeneous cultures, and these cells are disaggregated cells. So the current terms or the classification is organ culture, cell culture, primary explants, ex vivo propagation, and based on the composition, the cultures can be homogeneous and heterogeneous. So why do we need tissue culture? The main use of tissue culture currently and earlier was also research. It overcame the problems in studying cellular behavior, the confounding effects of the body and the subject, and the variation in animals due to experimental stress. So these were minimized because they were in a controlled laboratory environment. The animal use, the ethical aspect of animal use was also overcome. So it reduced the animal use. Then it also helped us in commercial large-scale production, like we saw in cervical cancer vaccine. Now the advantages were we had absolute control of the physical environment. There was homogeneity of the sample, and less amount of tissue was needed to produce more. The less compound was used and more results were seen. Now what were the disadvantages? It was a highly skilled work. It still is a highly skilled work. So it needs highly skilled and trained labor. The conditions are hard to maintain, so everything needs to be standard and up to mark. And there is again the cost factor is also high, and there is a phenomena of de-differentiation in which the cells lose their specialized ability, and they again go to their resting stage. So this and instability, which was molecular instability at the DNA or RNA level when they were undergoing differentiation, and aneuploidy. So organ culture and tissue culture. Organ culture is basically the entire embryo or organs are excised from the body, and a small part of the organ is taken and it is cultured. There is a recombination of different cell types, and a three-dimensional lifelike organ is tried to be cultured in the laboratory environment. Now the advantages are the normal physiological functions of the tissue are maintained and cells remain fully differentiated. Disadvantage is that the growth is slow and commercial production is not possible. In tissue culture, only the fragments of excised tissue like biopsies are used, and they are grown in culture media. Basically, tissue culture exploits two properties. One is proliferation, other is differentiation. So when there is the need of large scale production, the property of proliferation is more focused upon. But when there is the research of normal interactions and physiology and effects are to be seen in vitro of whatever happens in vivo. Lifelike conditions, for that differentiation is used. So organ culture focuses more on differentiation and tissue culture focuses more on proliferation. So in tissue culture, mainly growth is more important, and in organ culture, formation of a organ-like structure is more important. Now cell culture. Cell culture is basically just like tissue culture, only the amount of the tissue is less than 1 millimeter of the biopsy is used, and individual cells are used to create more cells. Now cell culture is of two types: primary cell culture and established cell lines. So primary cell culture are tissues and organs which are excised directly from the tissue by biopsy from a living organism, and they retain the natural function and all the natural properties of the cells, and then they are used to further produce or culture further cells. Now in established cell lines, the biopsy or the primary explants which are taken are from the tumors. Now these cells have a infinite ability to proliferate and divide. So they have or we can also produce established cell line by using primary cells also. So the primary cells which are normal, in them mutations are induced by exogenous phenomena like altering their DNA function or giving them any carcinogenic toxins, because of which they will undergo mutation, and then they will be transformed into continuous cell lines. And Hayflick limit. So this is the point at which the primary cultures stop growing or they undergo replicative senescence. As we all know, all the normal cells, apart from stem cells, they have a limited amount of growing potential. The Hayflick limit is basically based on the property of telomere shortening. So with each division, each cell division, the telomere in the chromosome, it shortens. And when the telomere has shortened to a certain limit, the cell cannot divide. It will undergo senescence. So this Hayflick limit is basically a problem in established cell lines. So for established cell line, we need either tumor cells or stem cells or totipotent cells which have the ability to proliferate infinitely. Now the types of cell culture are histotypic or organotypic. These two types are based on the morphological arrangement of the cells. So histotypic cell culture is basically when a single layer of cell is produced, and on that we produce multiple layers and we replicate a normal histologic structure of a tissue, and it is two-dimensional. So we give perfusion, we give substrate, and many layers are produced. In organotypic, three-dimensional production of the organ is tried. So the substrate will be more three-dimensional. Only one layer of the substrate is not used, and perfusion through exogenous substances is tried for those organotypic cell cultures. Now primary culture. It is the first isolation of the cell from the tissue before we use it for culture. So it has 4 stages: acquisition of the sample, isolation of the tissues, disaggregation of the cells from the tissue, and culture or seeding of the cells into the vessel. Primary explant is the suitable small amount of tissue which is used for attachment with the substrate. And primary explant, the substrate, basically the cells and the substrate interact with each other. So in a vessel, we need the cells to stick to the vessel wall. For that, we need a substrate. This substrate is taken similar to the ground matrix which we see in our body. So it can be plasma clots, fibrinogen, thrombin, type 4 collagen in 3-dimensional models. And disaggregation is nothing but mechanical or enzymatic separation of each and every cell after we have taken a small biopsy or the tissue from an organ for our culturing method. So disaggregation, most commonly used enzyme is warm trypsin, which is at 37 degrees Celsius and for 30 minutes. Now 30 minutes is important because the cell will get damaged if it's kept for too long. Mechanical methods are used to reduce proteolytic damage if we want to study mainly the chemical functions of the cells. So it can be scraping, sieving, syringes, spillage, or the very basic titration by pipette. Secondary culture is isolated by selection or cloning of the primary cell culture. It is also having finite lifespan, and it retains a differentiated phenotype. So it will resemble the primary cell. And this is a schematic representation of the primary culture and the continuous cell lines. So as we can see, here we have taken an organ. Now we have isolated cells by mechanical or enzymatic methods, and then we are making a primary culture of it. Now from this we have grown more cells. Now this is again subcultured, and then there are many generations of subcultures. But this division has a finite limit. So it will at a certain point undergo senescence. Now here the organ is having a tumor, and from that tumor we have taken cells, or we have taken normal cells and we have made them into tumor cells by inducing mutations. Now this has infinite potential of divisions. So this can keep growing. The culture media, it comprises of appropriate sources of energy and compounds which regulate the cell cycles. So it includes amino acids, vitamins, inorganic salts, glucose and serum. So biological fluids which are used are plasma, lymph, amniotic fluid and serum. But it was observed that for many cells, the growth factors which are present in serum, sometimes they act as a differentiating factor. So if we do not want the cell to become differentiated and we want them to proliferate even more, these growth factors which are present in the serum inhibit it. So then they decided to produce a serum-free media, and this media was made by specific labs, and each lab, they have their own set of serum-free media which are complex media. Other tissue extracts which were used as media was liver, spleen, tumors, leukocytes, bone marrow, the hematopoietic stem cells, chick embryo or bovine embryo. Clots which were used were coagulants or plasma clots. Balanced salt solutions, Eagles and Hanks, basically these contain sodium bicarbonate, sodium pyruvate and such balanced salts which are used as a substrate, and they help in cell to cell interaction at the substrate and the interface level basically. Basal media, MEM, which is minimally essential media, and DMEM, which is Delvecco, which was a scientist who worked on it. So he modified the media, he increased the amount of glucose and decreased the amount of sodium salts, and he produced the MEM media. And these complex media further were developed by laboratories, and they were customized according to their needs. Now stages in culture, as we saw, isolated tissue, primary cell culture which has a limited lifespan, finite cell cultures, and continuous cell lines. If we wish to produce continuous cell lines, then by inducing mutations, continuous cell lines. Now morphology of cells which we see in cell culture. Cells can be divided into three basic types based on their shape and appearance in cell culture: fibroblastic, epithelial and lymphoblast. So fibroblastic cells will appear bipolar, elongated, and they grow attached to the substrate. In epithelial, the cells are polygonal and they also grow attached to the substrate, but there will be a pattern of growth seen in the epithelium. And lymphoblasts, these are spherical in shape, usually in suspension. So they do not attach to the vessel or to any substrate. They will grow in a suspension. Now nomenclature of these continuous cell lines. There is to increase the uniformity, standardizations have been made. So specific codes or designations are given to the cell lines. NHB 2-1 represents the normal human brain NHB followed by cell strain 2 and clone number 1. Each cell line is designated, which is unique to avoid confusion. And the cell line should be prefixed with a code designating the lab in which it was obtained. So for National Cancer Institute, it was NCI. For Vistar Institute, it was WI. Now, examples of established cell lines. These are already present in use and which include HeLa, which was cervical cell cancer, then 293T, which is again from humans, and so on. Now, uses of tissue culture, cell culture, and organ culture. Mainly in screening of anti-cancer drugs, the effect of anti-cancer drug in vivo can be studied in vitro, and the effects or the ill effects, the good effects and the ill effects both can be studied. Then bioassays of cells can be made. Cytotoxicity of certain drugs or elements which we see. Then in vitro screening of drugs again. Production of antiviral vaccines, cancer research, cell fusion techniques, genetic manipulation, study of function of nerve cells, and chromosome analysis for cells derived from. Here are few examples. So epithelial cell culture. So this was an experiment in which gastrointestinal epithelial cell cultures were studied from murine colon, and the functions which were studied were cell-to-cell interactions, cellular microsignaling, and microbiological infections. They were also used in toxicological studies. In hepatocyte tissue culture, the hepatocytes, since they play an integral role in metabolism of drugs and nutrients, the physiology of these metabolisms was studied, drug interactions were studied, and the differentiation of HCC using flow bioreactor was also studied. Another experiment was of nervous system tissue cultures, and this was mainly focused on the functional aspect of the nervous system. So cortical-like tissues were reconstructed and the effects of drugs on these tissues were studied. And neural cell therapy was also experimented. The three-dimensional collagen gel culture was used for neuronal stem cells and progenitor cells to study the central nervous system stem cell development. Then type 2 pneumocytes and ARDS. So a lifelike condition was created where there was a media and the substrate interface, and in that interface the type 2 pneumocyte interaction in cases of acute respiratory distress conditions were studied. So the conditions were created as if the tissue is undergoing in respiratory distress, and this was quite helpful in understanding the basic mechanisms of ARDS. Then three-dimensional models. Three-dimensional models can be divided into normal tissue models and rotating vessel wall models. So rotating vessel wall model is nothing but the tissues are placed in a rotating vessel wall and they are agitated. So because of the agitation, they aggregate and they become three-dimensional. So that was only the different thing about rotating vessel wall models. So in this diagram we can see below is the assembly of the rotating vessel wall and the series of events, and above image we can see how the cells, after being agitated, they aggregate and they form then ground substance and three-dimensional structure is formed. Here are few microscopic images of the tissues which were cultured. So on the far left is the two-dimensional image in which we can see human lung cancer cells grown, and in the image next to it we can see the three-dimensional portion of the alveoli of the lung, and in the far right we can see the HNE stained sections of the cultures. So what is the take home message with tissue culture? It has helped us make great progress in research. It has helped us in studying normal physiology, interactions, exogenous substances, and with the help of continuous cell lines, we were also able to commercially produce a lot of things like vaccines and animal involvement. Like earlier, when we used to inject the mice with tumor cells and see the progression, that is reduced. However, it still remains mostly limited to research, and cost is also a great limitation for tissue culture. Again, organ culture also, they cannot be commercially produced in large numbers because first they go senescence, and second, sometimes if the adequate perfusion is not there, then they undergo hypoxic damage and tissue necrosis. These are my references. Thank you.